Hello, everyone. Welcome back to Meds.Concepts. Today, we are going to discuss one of the most important and high-yield topics of molecular biology, mutation and mutagenesis. Mutation refers to permanent heritable alteration in genetic material. These changes may occur at DNA sequence level, chromosome structure level, or chromosome number level. Mutations are extremely important biologically because they are the raw material for evolution, responsible for genetic diversity, associated with inherited diseases, and involved in cancer development. In this lecture we will study causes of mutations, physical and chemical mutagens, point mutations, frameshift mutations, suppressor mutations, functional mutations, Ames test, and mutation analysis in detail. So let's start with introduction to mutation. Mutation is defined as a permanent heritable change in the genetic material of an organism. These changes may occur in nucleotide sequence, chromosome structure, or chromosome number. Mutations can be beneficial, harmful, or neutral. Beneficial mutations provide adaptive advantages and contribute to evolution. Harmful mutations may lead to genetic disorders, metabolic defects, or cancer. Neutral mutations usually do not affect phenotype significantly. Mutations generate genetic variability, which is essential for natural selection and evolution. At the molecular level, mutations alter genotype and may alter phenotype. Causes and molecular basis of mutation. Mutations may arise spontaneously due to endogenous cellular processes. One major cause is replication error. During DNA replication, DNA polymerase may insert an incorrect nucleotide. Another important cause is tautomeric shift. Nitrogenous bases occasionally shift into rare tautomeric forms, leading to abnormal base pairing. Depurination is another spontaneous process in which purine bases, adenine or guanine, are lost from DNA, producing apurinic sites. Deamination is also common. Examples include cytosine converting into uracil and 5-methylcytosine converting into thymine. Oxidative damage produced by reactive oxygen species can also modify DNA bases. For example, atoxoguanine pairs with adenine instead of cytosine, producing mutations. Most spontaneous mutations originate during DNA replication. Spontaneous versus induced mutation. Mutations are broadly classified into spontaneous and induced mutations. Spontaneous mutations occur naturally without external mutagens. They result from replication errors, spontaneous chemical changes, and endogenous metabolic reactions. Induced mutations are caused by mutagens. Mutagens are agents that increase mutation frequency. Mutagens are classified into physical mutagens, chemical mutagens, and biological mutagens. Induced mutations occur at much higher frequencies than spontaneous mutations. Physical mutagens. Physical mutagens mainly include ionizing and non-ionizing radiations. Ionizing radiations include X-rays, gamma rays, and radioactive particles. These radiations generate free radicals that damage DNA. Major effects include single-strand breaks, double-strand breaks, chromosomal rearrangements, and base damage. Non-ionizing radiation mainly includes ultraviolet radiation. UV radiation induces cyclobutane pyrimidine dimers, especially thymine dimers. These dimers distort DNA structure and block replication and transcription. If unrepaired, error-prone repair mechanisms introduce mutations. UV-induced CC2TT mutations are characteristic of skin cancer. Chemical mutagens. Chemical mutagens alter DNA structure chemically. Base analogues resemble normal nitrogenous bases. 5-bromorycyl is an analogue of thymine and can pair abnormally with guanine. 2-aminopurine is an adenine analogue. Alkylating agents transfer alkyl groups onto DNA bases. Examples include EMS and MMS. Alkylation changes base pairing properties and produces mutations. Nitrous acid acts as a deaminating agent. It converts adenine into hypoxanthine and cytosine into uracil. Hydroxylamine specifically modifies cytosine. Intercalating agents insert between adjacent DNA bases. Examples include ethidium bromide, acridine orange, and proflavin. These agents mainly induce insertion or deletion mutations leading to frameshift mutations. Transition and transversion mutations. Base substitution mutations are classified into transitions and transversions. Transition mutation involves purine replacing another purine or pyrimidine replacing another pyrimidine. Examples include adenine changing to guanine or cytosine changing to thymine. Transversion mutation involves purine replacing pyrimidine or pyrimidine replacing purine. Examples include adenine changing to thymine or guanine changing to cytosine. Transitions occur more frequently because they cause less structural distortion in DNA. Base analogues commonly induce transition mutations. Point mutations. Point mutations involve alteration of a single nucleotide pair. 3 major classes exist: silent mutation, missense mutation, and nonsense mutation. In silent mutation, codon changes but amino acid remains unchanged because of degeneracy of the genetic code. In missense mutation, a different amino acid is incorporated. Missense mutations are of 2 types: conservative and non-conservative. Conservative missense mutation replaces an amino acid with a chemically similar amino acid. Non-conservative mutation replaces it with a very different amino acid, greatly affecting protein structure and function. Nonsense mutation converts a normal codon into a stop codon. This causes premature termination of translation and production of truncated proteins. Frameshift mutations. Frameshift mutations occur due to insertion or deletion of nucleotides not in multiples of 3. Because codons are read in triplets, such mutations shift the reading frame downstream of the mutation site. As a result, amino acid sequence changes completely, premature stop codons may appear, and non-functional proteins are produced. Frameshift mutations are generally severe. Intercalating agents are such as ethidium bromide, proflavin, and acridine orange commonly induce frameshift mutations. Classification based on cell type. Mutations are also classified according to the type of cell affected. Somatic mutations occur in body cells. These mutations are not inherited and affect only the individual. Somatic mutations are very important in cancer development. Germinal or germline mutations occur in reproductive cells. These mutations are heritable and transmitted to offspring. Only germline mutations contribute to evolution because they enter the gene pool of future generations. Mutations may also be inherited from parents or acquired during an individual's lifetime. Forward, reverse and suppressor mutation. Forward mutation converts wild-type phenotype into mutant phenotype. Reverse or back mutation restores mutant phenotype back to wild-type. True reversion restores the original nucleotide sequence. Suppressor mutation is different. In suppressor mutation, second mutation compensates for the effect of the first mutation without correcting it directly. Suppressor mutations are of two types: intragenic suppressors and intergenic suppressors. Intragenic suppressor occurs within the same gene. Intergenic suppressor occurs in a different gene. A classic example is suppressor tRNA mutation, where altered tRNA recognizes stop codons and inserts amino acids, suppressing nonsense mutations. Conditional, leaky and functional mutations. Conditional mutations express mutant phenotype only under specific environmental conditions. A famous example is temperature-sensitive mutation. At permissive temperature, protein functions normally. At restrictive temperature, protein becomes non-functional. Leaky mutation or hypomorphic mutation results in partial loss of function. The gene product still retains some activity. Functional mutation classes include amorph, hypomorph, hypermorph, neomorph, and antimorph. Amorph mutation causes complete loss of function. Hypermorph mutation increases gene activity. Neomorph mutation produces a completely new function. Antimorph mutation produces dominant negative effects. Gain and loss of function mutations. Loss of function mutations reduce or abolish gene activity. These mutations are usually recessive because one normal allele can often compensate. Gain of function mutations increase gene activity or produce novel functions. Gain of function mutations are usually dominant. Dominant negative mutations are special gain of function mutations in which mutant proteins interfere with the function of normal proteins. Gain of function mutations are highly important in oncogene activation and cancer development. Lethal and polar mutations. Lethal mutations cause death of the organism. Dominant lethal mutations kill even in heterozygous condition. Recessive lethal mutations become lethal only in homozygous condition. Polar mutations mainly occur in prokaryotic operons. In operons, multiple genes are transcribed together as polycistronic mRNA. Mutation in one upstream gene may affect expression of downstream genes. Nonsense mutations commonly produce polarity effects because premature stop codons terminate translation early. Ames test and mutagen detection. The Ames test was developed by Bruce Ames to detect mutagenic potential of chemicals. The test uses mutant strains of Salmonella typhimurium that cannot synthesize histidine. These bacteria are called His- strains. If chemical induces reverse mutation restoring histidine synthesis, His+ revertants appear and grow on histidine-deficient medium. The number of revertant colonies indicates mutagenic strength of the chemical. Rat liver extract is often added because some chemicals become mutagenic only after metabolic activation. Ames test is important because many mutagens are also carcinogens. Finding nature of mutation by reversion analysis. Different mutagens preferentially induce specific mutation types. By studying reversion frequencies produced by different mutagens, scientists can determine the nature of unknown mutations. For example, EMS commonly induces GC2A transitions, 2-aminopurine induces AT2GC transitions, nitrous acid causes deamination, and ethidium bromide induces frameshift mutations. Reversion analysis is therefore useful in mutation characterization and molecular genetics research. Finally, let us revise the most important concepts. Mutations may be spontaneous or induced. UV radiation produces thymine dimers. Ionizing radiation causes DNA strand breaks. EMS is an alkylating agent. Nitrous acid causes deamination. Intercalating agents induce frameshift mutations. Transitions occur more frequently than transversions. Nonsense mutations generate stop codons. Germline mutations are heritable. Suppressor mutations restore phenotype without correcting the original mutation directly. Leaky mutation is also called hypomorph. Hypomorph mutations produce gain of function. Polar mutations affect downstream genes in operons. Ames test detects mutagenic chemicals. Strong mutagens are often carcinogenic. Thank you. If you found this video helpful, please like, subscribe and share with your friends.